Bitter criticism. Reading news. This will be a more regular content because there's interesting facts about science, and there's interesting facts actually. There's two discoveries. Scientists just discovered a fungi. I don't know how to spell fungi. a fungus that eats plastic. I have seen a lot of publishing on scientific media, on Instagram. Fungus eats plastic. Fungus eats plastic. News. There's a lot of like Times of India. Maybe there's a lot of fungus that eat plastics. I'm going to read this news from this article from Times of India, and says, Times of India, plastic eating fungi. They said fungi could help clean up oceans, say scientists. TOI, Science Desk, timesofindia.com, July 22. There's a photograph, a lot of plastic bottles on the sea. German researchers have identified freshwater fungi capable of breaking down synthetic plastics, offering a potential natural solution to plastic pollution. These fungi thrive by consuming certain plastics, particularly polyurethane, though they struggle with tougher plastics. I guess this is all the same, but I have more publications on Instagram from science pages. While not a complete solution, scientists believe this fungi could complement recycling efforts, especially in controlled environments like wastewater plants. Interesting, isn't it? How a fungi, how anything can eat plastic. So fungi are eating plastic. What about that? A surprising discovery by German researchers has identified fungi capable of breaking down synthetic plastics, potentially offering a natural solution to one of the planet's biggest environmental threats. These plastic-eating fungi, found in freshwater habitats, can survive by feeding solely on certain types of plastic. While not a cure-all, scientists believe this could be a valuable tool in efforts to clean up plastic waste shocking the world's oceans. Their ability to digest plastic without additional nutrients sets them apart from the organisms studied for plastic degradation. How ocean-dwelling fungi could become allies in the plastic pollution crisis. In Lake Stechel, Germany, scientists at the Leibniz Institute of Freshwater Ecology discovered that several strains of microfungi could grow entirely on synthetic polymers. These fungi were not only surviving, they were thriving, forming biomass without any other carbon source. This adaptation likely emerged in response to the abundance of plastic in aquatic environments. Notably, 4 of the 18 fungal strains, studies showed a strong appetite for polyurethane, a common plastic used in building materials and foams. However, they were less effective against tougher plastics like polyethylene used in bags and wrappers, and tire microplastics, which contain metal additives that hinder decomposition. While the idea of developing these fungi directly into the oceans is appealing, researchers caution that their plastic degrading enzymes rely heavily on factors like temperature and nutrient availability, making them far more efficient in controlled settings such as wastewater plants or industrial sites than in open seas. And there's more. Why fungi aren't the silver bullet but still matter? I'm going to compare all this to my Instagram publications. Despite their promise, scientists stress that fungi alone won't solve the global plastic crisis, with plastic production surging to 400 million tons annually and only 9% recycled globally. More synthetic changes are needed. The fungi's role may be best suited to complement traditional recycling and waste management, particularly where existing systems fail. Plastic degrading organisms have long fascinated researchers. Over 400 species of fungi and bacteria have been identified with plastic eating potential. Other solutions being explored include quote-unquote self-digesting plastics embedded with bacterial spores. Yet most of these biological solutions face the same challenges: slow breakdown rates and strict environmental requirements. The discovery adds momentum as global leaders negotiate the first international plastics treaty, with the final phase of talks set for later this year. Experts hope that combining innovations like these fungi with strong policy will turn the tide on plastic pollution before it overwhelms ecosystems entirely. Yeah, and that's it. I'm going to compare this with what I saw on Instagram. A lot of things, and even Facebook. I guess I saved something on Facebook. Look. Very interesting news from, look, this is in the Amazon actually, so it's not the same research. I guess not the same. I'm facing trouble here on regular Facebook. I will try the Facebook Lite. Facebook Lite. It says humor. This is a humor page, a page called Humor on Facebook. Scientists have found a fungus in the Amazon rainforest called Pestalotipes, something like that. Pestalotipes microspora that can naturally break down polyurethane plastic. This fungus even thrives without oxygen, making it unique. Discovered in Ecuador's Yasuni National Forest by Yale student researchers in 2011, the fungus uses plastic as its only food source. It fully digests the plastic through enzymes, instead of just breaking it into smaller pieces. This discovery has sparked global interest as a new way to fight plastic pollution sustainable to fight plastic pollution sustainably. I guess it's not the same experience. And there's no article. There's no article here. What about this? No article on. Yeah, humor has no article, just a regular publish. I guess. I guess I'm done. I'm done with this news, but I'm going to try on. But this has been the last week, you know, not. This has been last week, so I'm not sure if it's still available on my stories on Instagram. Let's see. Plastic eating. Fungus eating plastic. Where is it? Where is it? So I guess that's it. Well, the next one is about Down syndrome. Oh, there's this is actually news. Look, they are. I don't remember how to say this. This is a regular plant. You can eat the roots of this regular plant and survive stomach cancer. There's cure for stomach cancer by eating regular root plants. Again, I don't know where I left this. I'm going to research on I'm going to Google it. Crisp Down syndrome. So looks like Down syndrome is done. Down syndrome will be over. There will be no more Down syndrome people. What about that? There's a news from Azerbaijan. What about Azerbaijan? News.az International. Scientists claim to remove chromosome linked to Down syndrome. Yeah, that's the news. Using CRISPR or something. July 2022. Japanese researchers from Mie University have developed a groundbreaking gene editing approach that may eliminate the extra chromosome responsible for Down syndrome, according to findings published in PNAS Nexus. Down syndrome, the most common chromosomal abnormality, is caused by an extra copy of chromosome 21, trisomy 21. Using CRISPR Cas9 technology, the scientists successfully removed the surplus chromosome from lab-grown cells, restoring normal gene expression. Yeah. CRISPR, known for its DNA cutting capabilities, was programmed to precisely target the extra chromosome through a technique called allele-specific editing. News.az reports, citing foreign media, follow-up experiments revealed significant changes in gene activity. Genes linked to nervous system development became more active, while those associated with metabolism slowed down. The treated cells also exhibit faster growth compared to untreated ones. Faster growth compared to untreated ones. Nice. So yeah, Japanese people, they defeated Down syndrome. What about that? The team extended their testing to skin fibroblasts from individuals with Down syndrome, achieving similar results. However, researchers caution that the method is not yet ready for clinical use due to potential off-target effects on healthy cells. Work is ongoing to refine the process for greater precision and safety. Looks like Azerbaijan is a rich country. So nice, you know, there's zero chance if I will have if I will have children, there's zero chance of have a Down syndrome kid. Such a bless. I'm sorry, but Down syndrome is terrible, I guess. I'm going to read another one. A pretty old one. This is very old. This is from February 23. February 23. Very old one. And it says, look, there's a lot of dogs here. And the dogs just leave me. A beautiful pit bull. And looks, my friends are coming. But I guess they will not talk to me. I don't know why. They are great people. They do know what they could do. They could do like party, and they could make a company of party. Do you want a party? Call them. They're always partying. Anyway, there's a news from SCI Tech Daily, ScienceTechDaily.com. This is very old. It's from February 23, like 1 year ago. Like 6 months ago. CRISPR snips away extra chromosomes, offering new hope for Down syndrome treatment. Thanks to Japanese people. This article is by PNAS Nexus, February 23. A short text of 2 minutes. There's a photograph. A proof of concept study shows that CRISPR Cas9 can eliminate extra chromosome copies in Down syndrome cells. The method restores normal gene functions, but is not yet ready for human trials. Scientists are exploring gene editing as a way to correct trisomy at the cellular level using CRISPR-Cas9. Researchers successfully remove extra copies of chromosome 21 in Down syndrome cell lines, restoring normal gene expression. This breakthrough suggests that with further development, similar approaches could be applied to neurons and glial cells. Oh, look at that. Neurons and glial cells. I wonder if this is there will be a therapy. You know, maybe people will never die. Look at me. I lost a foot and I'm still alive. No, I'm not. I didn't lost a foot, but I'm recovering. You know. Oh no, you will not have glycophone. I will read again. I lost. Scientists are exploring. With further development, similar approaches could be applied to neurons and glial cells, offering a potential treatment for those with the condition. Gene editing for trisomy treatment. Scientists are exploring gene editing as a potential way to treat trisomy at the cellular level, according to a proof-of-concept study conducted in lab-grown cells. Down syndrome occurs when a person has an extra copy of chromosome 21, affecting about 1 in 700 live birds. While the condition is easily diagnosed early in development, no treatments currently exist. Yeah. No treatment, so people can diagnose but cannot treat such a bad luck. In this study, Ryotaro Hashisume and colleagues used the CRISPR-Cas9 gene editing system to remove the extra chromosome from trisomy 21 cell lines. These cells were derived from both pluripotent germ cells and skin fibroblasts. The technique was able to identify precisely target the duplicated chromosome, ensuring that after removal, each cell retained one copy from each parent rather than two identical copies. And there's a graphical abstract summarizes the approach. And we have this is very technical, allele specific CRISPR-Cas9 chromosomal rescue for trisomy 21. By suppressing the cell's natural DNA repair mechanisms, researchers increase the efficiency of removing the extra chromosome. Their findings show that this process restores normal gene expression and cellular function in edited cells. Potential for future medical interventions. Although promising, the method is not yet ready for use in living organisms, as it can also alter the remaining chromosomes. However, researchers believe that similar approaches could eventually be applied to neurons and glial cells, paving the way for potential future treatments for people with Down syndrome. Reference, there's an article, quote, trisomic rescue via allele-specific multiple chromosome cleavage using CRISPR-Cas9 in trisomy 21 cells. That was by Ryotaro Hashisume. Maybe a medicine Nobel Prize, isn't it? I will see if Ryotaro is going to win the Nobel Prize. I will say Ryotaro Hashisume Nobel Prize. Not yet, I guess. No, not yet, I guess. But in my opinion, there could be nothing. I like Down syndrome people, but such a hell to be down. And well, the last one, I don't remember the last one. Down syndrome. Oh, I forgot. I'm not a Down syndrome, but I just forgot. I'm going to see. I guess I forgot everything. There was a old news now saying that there will be no more death, and there's this fungus and stuff. I'm going to try to again on stores to try to remember. There was an interesting one. Oh yeah, this look, this cancer on stomach cancer. Stomach cancer prevention, treatment, stomach cancer treatment, eating roots, eating roots of plants. Well, there's a lot of material, but what I saw is that this is a very popular plant. This is like an invasor species called, in Spanish you say diente de león. Well, in Spanish you say well, you say diente de león, Taraxacum officinale, Taraxacum officinale. Look, I have to research all that. Taraxacum officinale, Taraxacum officinale, stomach cancer. It's not the plant but the root. Look, dandelion, you know, dandelion was harvested, and this is very old, 2017. National Institute, look, dandelion root extract suppress gastric cancer cells proliferation. That's nice for all those who have stomach cancer. Dandelion root extract suppress gastric cancer cells proliferation and migration through targeting ENC RNA CCAT1. Juan Zhu et al. Biomed Pharmacotter, 2017 September. Abstract. This is a science article. Gastric cancer, GC, is one of the most common tumors worldwide. Standard treatment after early detection involves surgical excision. Recurrence is possible, and metastatic gastric cancer is refractory to immunoradio and most harmful chemotherapies. Various natural compounds have shown efficacy in killing different cancers, albeit not always specifically. In this study, we show that dandelion root extract, DRE, DRE people, such a fancy name for dandelion roots. There'll be a lot of dandelion everywhere. There's a plague, dandelion plague. Specifically and effectively suppresses proliferation and migration in human gastric cells without inducing toxicity in non-dangerous cells. Long non-coding RNAs, INC RNAs, are known to promote tumorigenesis in many cancer types. Here we show that the INC RNA colon cancer associated transcript 1, CCAT1, was downregulated in dandelion treated GC cells. Furthermore, downregulation of CCAT1 inhibited proliferation and migration of gastric cells. We also found that DRA exerted its functions in GC cells partially through targeting CCAT1. This data will provide a basis on which further research in cancer treatment through DRE can be executed.